Thank you to the audience for your patience while we got set up there. And a big welcome back to James from Mindset Pharma and Joseph Araujo, one of the premier drug development companies in the space. James, I'll let you do a little bit of an intro, and I'll hop back on when it's time for audience Q&A here.
Awesome. Thanks, Patrick. And thanks for everybody for tuning in to watch our presentation. It's great to be back here at Microdose. We were on a little while ago, and so I think our intention tonight isn't so much to give you the full mindset investor presentation or pitch, but we wanted to use the time as really an opportunity to update you, to update the community on where we're at on our new drug development. So this is going to be a lot more about Joseph than it is about me talking about mindset. But just to kind of just quickly to refresh you on what mindset is. So we're a biotechnology company with two focuses. One of them is on developing next generation psychedelic drugs, so novel chemical entities. These are drugs that are, broadly speaking, are inspired by psilocybin or based off of psilocybin. And we think new drugs are lots of debate over this point, but in our view, new drugs are really critically important because we think, really simply, there's an opportunity to improve, on the classic drugs, and not only in terms of toxicity, but in terms of their effectiveness and their predictability and their duration. And we think that if we're successful at making drugs that frankly are more convenient for physicians and for patients, the adoption is going to be that much greater. So that's fundamentally why we think new drugs are important. We also, the other side to our innovation is a chemical synthesis program to synthesize psilocybin as well as our own compounds. As everybody knows, access to GMP grade psilocybin is expensive today and not straightforward. And so there's a few groups doing great work out there, like Scigen, but we've come up with a very straightforward chemical synthesis process that we believe allows us to synthesize psilocybin at a fraction of the cost available in the market today, and at a very large scale. So we're very excited about that. Today, we're really going to focus mainly on the NCE program. And the NCE program, we started with the goal of really trying to design a next generation psilocybin, and that was really for a couple of reasons. Number one, it was to us the drug that had the most evidence and the most regulatory momentum for the widest possible indications. So it could help. The greatest MDMA is a tremendous drug, but psilocybin could help a greater diversity of people. And as much work has gone on in psilocybin, most of that has not gone to the level of drug design, or it hadn't, at least until relatively recently, to understand whether, given our understanding of serotonin receptors and how the drug interacts with the brain, whether there was an opportunity to use that understanding and to create something better. And so our program really has expanded quite a bit from where it started off with. And so at this point, we have, and Joseph will get into this in more detail, we've got a whole range of different acting compounds that embody the same the same psychedelic experience as psilocybin, but do so through what we think is a more optimized chemical structure, and we think that will have real benefits to patients. We think that there's a lot of noise about drug development and new drugs in the psychedelic space, but we think that, respectfully, we are really one of the early leaders in this space, in this particular area of the space, at least based on the date of our patent filings, the level of preclinical data that we've been able to generate, and simply the range of different potential drug candidates that we are now moving forward with. So I'll turn it over to Joseph, really, to walk you through what we saw as in a little more detail as some of the opportunities to create a better drugs, and then where we've gotten to so far.
So thank you, James, and thank you to the organizers. And it's great to follow up with so many innovators, and as people have said earlier, we're sort of standing on the shoulders of giants here. And my background has been in preclinical development and animal models for predicting clinical outcomes. And as James mentioned, psilocybin is particularly interesting. There's an enormous amount of data. And in starting the company, you kind of want to focus on things that you're good at. And we brought in Malik Slassi, who's our chief chemist, who's a phenomenal chemist, who's really expanded what we started trying to do, and now we have a lot larger platform that we're working from. So when we first started looking at the space, and really our goal was to focus on providing something for people who need it most. So patients with hard to treat diseases. And it was clear to us that mushrooms in and of themselves probably wouldn't fall into that market due to differences in purity. One can extract and create a consistent psilocybin product, and clearly that's been very positive in early clinical trials and lots of data in the past. But with this patient population, you're looking at polypharmacy. There's several metabolites, and it's still really, in the grand scheme of things, it's still early days in understanding how these compounds behave in different populations. And so, there could be fast and slow metabolizers, and time will tell that. So our goal was really initially to focus on psilocybin analogs, really follow what people were creating in the clinic, and have follow-up molecules. And really that's extended quite a bit. And ultimately, we felt that patent protected molecules would be important to help really interrogate the science behind psychedelics, both preclinically and potentially to develop new drugs that would fit into different categories. So I think the two most common approaches that people are speaking about, and there's been lots of information here, so I won't spend too much time here, but really macrodosing, psychotherapy assisted macrodosing approaches, which will ideally be in new clinical settings and a new way to treat psychiatric illness. And we have some ideas on what indications that could work with. We hear about major depression and substance abuse disorders, but more and more there's a movement towards microdosing, or more traditional pharmacological approach, and ultimately a macrodosing approach where people have hallucinations may not be the right approach for everybody. And there's some particular indications where we believe that psilocybin and psilocybin-like drugs might be particularly beneficial. Pediatric populations, for example, with ADHD, where clearly one wants to avoid a hallucinogenic type of effect, or potentially an Alzheimer's disease. And so we see different drug profiles, or we believe there's different drug profiles for both, with microdosing potentially being a lower effect size in terms of effect and not leading to hallucinations, and then longer acting to ensure that potentially once a day or once every couple day dosing could be used. So at this time, we have three families that we've really been focused on, and we've been following the data that we're generating preclinically, primarily in vitro, and we've now begun our in vivo testing. So we won't speak much about that here, except we're seeing CNS penetrant drugs and orally active drugs across all of our classes. But the data and also the chemical approach and scaffolds really seem to we can sort of frame the three families into potentially what indications and how they might become useful, and really continue to follow that data. So our first family was really intended to be psilocybin analogs, deuterated analogs, and prodrugs, which we felt could follow aspects of the 505(b)(2) pathway and really be quick, or molecules that could be quickly adapted for and enter clinical trials and be very much psilocybin-like drugs. So not potentially affording the benefit of having fewer metabolites and maybe having more consistent dosing, but ultimately being very psilocybin-like. Our second family is our restricted analogs, and what we're seeing there is a range of effect size when we look at 5-HT2A functional assays. So these potentially we see as next generation macro dosing type drugs, ideally with shorter half-lives, which we feel could benefit from in the clinic, where instead of having an 8-hour session with psychotherapy, one could potentially envision an hour or 2-hour session, which is similar to what's being described with drugs like DMT and 5-methoxy DMT. And our third family is also a restricted class, where we're seeing a lot lower effect size on receptor assays, functional receptor assays, but potentially the opportunity for longer half-lives. So I'll talk a little bit about some of the in vitro data across these classes, and once again, these are just exemplars to demonstrate how we're thinking about what we have. So on the left-hand side here, this is a functional 5-HT2A assay, and in red, we have psilocin, which is our reference compound in vitro. And you can see that we have compounds that are similar effect size. And on the far right, you can see from a receptor binding perspective, very similar to psilocybin. So this is MSP1002. And in the lower right-hand corner, we have in vitro ADMET data suggesting half-life a little bit longer than psilocybin in this case, but or psilocin, sorry, in this case. So once again, this family really potentially could follow a 505(b)(2) pathway and move quickly into the clinic based on the data that's available with psilocybin and some of those indications. Our second family, once again, if you focus on the left, you can see the red line there. Once again, that's the same as what we had in the previous slide. And we have several compounds that have a much larger effect on the serotonin 5-HT2A receptor. In some cases, 7-8 times greater. This potentially could be these could be great macrodosing targets. The exemplar here is MSP 2015, where you see similar receptor binding, a much larger effect size, with a similar half-life. So we're anticipating that we may be able to select some compounds from this class that are potentially, let's say, have that hallucinogenic and mystical experience effect that people talk about, but potentially with a shorter duration of action.